Howdy everyone, I'm one of your Blue Jeans and Boots hosts, Joe Romanowski, here to let you know that the Interdisciplinary Graduate Program in Genetics and Genomics at Texas A&M University is currently accepting applications for our PhD program. Whether you've listened to our podcast for a while or are new here and have been thinking about graduate school, check out our flyer posted in our website, genetics.tamu.edu. Our program is truly interdisciplinary and includes cellular and developmental genetics, evolutionary genetics, bioinformatics, plant genetics, medical and applied science, and so much more. Come learn from researchers in more than 20 different departments and find your scientific passion today. The application is free and the stipend is very competitive. Learn more on our website at genetics.tamu.edu. Now let's talk about some science. Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show to pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we will talk to Benjamin Thomas. Ben is a PhD candidate in the Interdisciplinary Graduate Program in Genetics and Genomics. His lab is in the Department of Microbial Pathogenesis and Immunology. I'm one of your co-hosts today, Joe.
And I'm your other co-host, Brittni.
And I'm Ben.
Well, thanks for coming, Ben. Ben, you want to tell us what you do in lab? What's your project like? What's your lab do?
Awesome. Thanks for having me. I'm in the Zhilei Chen's lab, Zhilei Chen. In that department that you mentioned, and we do a lot of protein engineering work. I guess trying to not replace small molecule drugs like, I don't have like examples off the top of my head, but drugs like penicillin, all the chemical-based drugs, we're trying to use proteins as a way to, another avenue for treating people with different, pathologies. Proteins, could be more bioavailable, have better pharmacokinetics within the human, and maybe have a better outlook on the patient's final outcome for treatment.
Okay.
We engineer these different proteins with different directed evolution methods to a target of interest. Some targets in the lab that we're interested in, SARS-CoV-2, Shiga toxin, Clostridium difficile, and those toxins that are secreted from that organism. And then, for my project specifically, I use them for diagnostics because not every drug makes it through the clinical trials. It has a lot of leaps and bounds and lots of funding to get and acquire and lots of stuff to get through in order to get to that finale. So I use the proteins for diagnostics. Point of care diagnostics, to be specific, because you can't just bring like a huge PCR block or some kind of this complicated lab equipment to places without a lot of resources. So that's pretty much the aim of my sector. But then there's like two other projects, one relating with AI, artificial intelligence, to just design de novo proteins that are trained on natural sequences that have evolved throughout the millions of years of evolution, and then use that algorithm to design new proteins that can treat different pathologies. And then another project just aiming at membrane proteins, with how valuable membrane proteins are to cell survival and other organisms' way of life. Just trying to create proteins, use DNA to create proteins that can target these different membrane proteins and have therapeutic benefits. But that's just like a somewhat brief overview.
So a lot going on.
Yeah. Too many cooks in the kitchen.
So it sounds like a lot of it is drug discovery and development for medicinal applications, some technology development going on too. You talked about your diagnostics that you're working on. There's a couple of things I think we need to elaborate on a little more. I have a couple questions about what's Shiga toxin and why do you guys work on that? We all know what SARS-CoV-2 is, like you mentioned, but
yeah. Okay. Well, Shiga toxin could be in a lot of undercooked foods. It's like burgers, steaks, maybe chicken. And also undercooked vegetables. They could be in the groundwater. It's from E. coli or Shiga toxin producing E. coli, deemed as STEC. That's the acronym. And I guess they got the machinery to encode for this Shiga toxin from, I guess, another bacteria called Shigella dysenteriae. A lot of stuff happening in the soil or other organisms, but if you think about where like the cows are eating or the pigs, they eat a lot of this, the ground, a lot of the grass, whatever they're fed with, that could be containing these microorganisms, and then they don't have receptors in their stomachs that, allow for the Shiga toxin to actually give. I guess when bacteria secretes the toxin, that toxin doesn't have the receptor to bind to actually give the pathology to that, the livestock, and so the livestock can poop, be fine, and transfer the microorganism because it's in its gut. It's just being pooped out, and then it can also get into its different organs or pass through its body. And so we're eating that meat downstream, and we can get a lot of those pathogens.
And we have the receptors?
Yeah,
and
we have the receptors.
Bad.
Yeah.
Oh, okay.
So within that 3-day period, maybe a week period of ingesting enough of that Shiga toxin, or the organism that can secrete it, you get diarrhea, and that's probably fine in a lot of people's cases. They're like, oh, it's whatever, it's chill. And then another couple of days, maybe a week later, you get bloody diarrhea. Then it's a question of, should I go to the doctor? Probably. And then if you go to the doctor and they don't really know what's going on, they need to prescribe you antibiotics, and those antibiotics pop open the bacteria that's producing the Shiga toxin. You get more of the Shiga toxin in your stomach and cause more of this this feedback loop that's happening. And then if you also have the antibiotics prescribed, maybe that helps with Clostridium difficile. I guess that's another stool-related pathogen. You can get the toxin that's causing you to have a lot of bad outcomes in the hospital.
And this is another one of your lab's research focuses.
Yeah.
Clostridium. Okay.
So you want to be able to diagnose or at least figure out which is, causing that issue. But yeah, back to Shiga toxin. That's a pretty bad issue. There's no authorized or approved medication for Shiga toxin, but it would be nice to know not to at least be prescribed more antibiotics, or at least watch what you're eating and maybe don't come into contact with someone else, because I think you can transfer it through fluids, but because it's in your stool, it could be in your bloodstream. I guess once the Shiga toxin gets into your stomach, it gets transferred into your bloodstream and then transferred to all your different organs. It hits your kidneys, your central nervous system, obviously your intestines, and then cause a lot of issues. But
so y'all try to target that Shiga toxin in some way to deliver a treatment in some kind of way?
Oh yeah, one of the novel techniques we're trying out, because drug delivery, you can make a drug, you can make a compound, but what matters is getting it to the target of interest. So in the lab, we are thinking about using lettuce, cabbage, as a way to transport whatever, because we have a DARPin right now that's able to neutralize the toxin and then save mice.
DARPin's one of your proposed delivery systems, right, that you're working on, your piece of technology that's supposed to deliver some kind of treatment. Yeah,
so DARPins are, we have antibodies that are in our bloodstream. They bind to stuff with high affinity. Antibodies are pretty big. They're very expensive to make. You make them in usually mammalian cells, which can be a lot of work, right? You have to need the media, the reagents, and all that tools, and specialized hands. With DARPins, I guess y'all can Google it later, but they're smaller versions of antibodies or synthetic scaffolds designed by somebody else to be pretty stable and commercially viable to make an E. coli. And we have one that can neutralize the toxin. And so delivery is the issue, because they can be degraded by your stomach's acids and stuff. And so I
was going to say, so it neutralizes the toxin but doesn't do anything to the E. coli producing it?
Yeah.
Okay, so it avoids the issues with antibiotics that would arise then. That's the goal?
Yeah, because you would, you would ingest it, and the toxin, the DARPin will go through and then bind to that toxin. The E. coli can keep secreting it, but the toxin at least will be neutralized, and the patient won't suffer as much.
So it seems like the delivery systems that y'all are working on can be applied to a lot of different pathologies, right? So it's pretty wide ranging. You mentioned Shiga toxin and C. diff is those are two potential applications, but I imagine that a lot of things, or a lot of different pathologies that you'd like to deliver some kind of medication in a specific way your type of technology would help with. What kind of got you into that for grad school? Because that's like a really like specific niche thing. It's like really applicable, but like how'd you get there from the genetics program into that lab?
So I guess when I was applying to the genetics program, I was like, you know what? I really like engineering and like medicinal stuff. There were some labs across campus that did that, so I picked Zhilei Chen's lab first, and then I didn't even know what project I was working on for that rotation. I was like, oh my goodness, what is this? There's a lot of DNA and nucleotides all over the screen and some proteins, and I was like, this is ridiculous.
It's like drinking water from a fire hose.
Yeah. We got there, though. We got there. But it's pretty cool stuff. I think my mentor is pretty, like, innovative. And then we have lab meetings every week, so everyone just throws ideas in there. Like the lettuce idea, I would never have thought of. Just putting, like, a DARPin within lettuce. Your stomach's acid doesn't digest cellulose, right? And then that can go through your intestines pretty well to eventually be broken up to release that DARPin in the right area, but...
I don't do any type of deliverable of like medication or any of these technologies that you talk about, at least with my work in the lab. So I'm really curious, like what goes wrong when you try to do these experiments? What do you think about when you try to design some type of deliverable drug or protein for Shiga toxin or C. diff or COVID or anything like that they all work on?
Are there any like off-target effects that are like really big ones that you kind of have to hurdle over before you use it for something?
So a lot of the, I guess, in vivo work, we have like collaborators that do that. So I guess from our side, what we have to worry about is the protein that we're creating reproducibly, like I guess the same? Is it as pure as the first batch? Whatever batch works, is it same? Because I guess you can purify the protein, but there could be all these other different fractions that come out that are just smaller, they're truncated, maybe from early translation, they got terminated early, or maybe the protein's unstable and it just starts folding and making these huge aggregates, which I guess is a lot of the inflammation that can be caused from protein drugs being injected into somebody. Maybe they're unstable and they just aggregate.
I imagine that's probably not there's probably a lot of labs or like whether they be commercial or academic labs that have similar issues. How do they try to overcome that?
I think early on, a lot of labs would just mutate certain residues.
Okay.
Pick an amino acid or two that's maybe responsible for stability, and then try to make sure it's soluble or it's like not aggregating in water, because I guess our blood is aqueous or water based. But I guess now people are using like machine learning to really, I guess, train the model on what's unstable, what's stable, and then create a better protein. So I'm like, I guess more of the rational design is falling out and more of the machines. But
you mentioned a while ago actually how you guys are using AI to try to develop these medicinal proteins. How does that even work? Do you like input large data sets and be like, hey, this is a data set from something that's already been done, and have it kind of sort out what's the correct protein and have it learn from like that previous data of how to select like potential like therapeutic proteins for your projects in the future? Like how does that even work?
So I guess for that's a good question. I mean, I guess for the grant I just applied for, the paper that we're basing this off of, or I guess the proposal, it was like from his Facebook group. They have this algorithm, I guess that's using language models, some kind of algorithm that's basically trained on maybe 100 million different proteins from like, I guess, different phylas, different classes, domains, and they're assembled them all, and they're like, okay, well, these are the most evolutionarily fit proteins over time. And then they told the algorithm, I guess they applied it to protein design. So you can basically use this algorithm online. It's like, I guess all you need is like to find the paper in this Google Colab notebook, and they basically provide you the Python code, and you put in like the primary sequence of the amino acid, whatever sequence you need, and then they'll recommend at each position what they think is more evolutionarily favorable. And a lot of these positions that they're recommending actually led to an increase in the activity. Like they had a couple of antibodies that were in the clinic already I guess, clinical phase trial 3, or maybe they were binding to Ebola virus, SARS-CoV-2 or flu virus, and they're able to mutate all of them with this algorithm that's just trained off of, I guess, evolutionarily fit proteins, and then they were able to recommend the correct mutations. And I think we compared the recommendations with our lab's proteins, and it was really interesting to see that whatever we were able to do in the lab and evolve our proteins, the same evolutionary, I guess the amino acids that evolved through the experiments were the same ones that the AI recommended too. So it was like this algorithm works in like 12 minutes. And
so it can kind of predict how the protein's going to evolve. And you guys are working in E. coli.
Yeah.
Right? So you try to like have E. coli express some type of protein that you think has therapeutic application. And over time, like as E. coli will replicate this, certain mutations might come up, and AI is able to predict those mutations and kind of give you your endpoint protein of what it should be.
Yeah, we can. I guess we make the libraries within the E. coli, or you can use degenerate primers. So you have like a sequence that's encoding for your protein. You can have these short strands of DNA that have, and then the companies that design these short strands of DNA, they can mutagenize or randomize each position, and for it to still anneal or bind on to that, I guess, still be used to amplify the sequence that's encoding for that protein. So then you can get all these different combinations of proteins, a huge library, and the AI was able to beat us to that position. Because this took like 6 months to like 2 years, and then this AI program took 12 minutes. So
That's a big difference. So with the AI, when you say evolutionarily fit, what types of conditions or like things are the AI using to determine what's evolutionarily fit? Is it having to do with like what types of proteins have conserved function over time? They've like acted the same way for a long period of evolutionary time, or are there certain functions it's looking for outside of that?
I think it's just evolutionary time. Like it doesn't take into account the target, whatever it's binding to, the affinities, like how strong does it bind. It doesn't take any information in, just what's lasted over the years. And that's like mind blowing just to think about.
So it's totally like, it's not specifically tailored to your context in particular. Like you can't like input parameters of like the, buffer or solution it might be working in, or the specific structure you want it to target or anything like that. It's just like based off of evolutionary data of how proteins have evolved in the past, it's kind of like trying to predict. Is that how it works?
Oh yeah, that's right on the money. That's
okay.
Still, I'm pretty scared of it myself, but
that's like the way a lot of things are. I mean, AI is being used more and more in the labs. Can even write some code. So, wow, ahead of the curve. You're right on the curve right now. So that's a pretty good segue into my next question. But what's like maybe one of the more exciting things that you've worked on in the lab? Like maybe a discovery or maybe a technology that you're working with. Yeah, just talk a little bit about that.
I guess for the Shiga toxin detection stuff, I guess that's the most exciting thing to me. That's the only thing that's been working the most.
Okay. So whatever's working is the most interesting.
If it's not working, it's not cool. Is that what you're saying?
I'm just like, man, pain.
We all relate to that.
Grad
It's
school.
like one thing doesn't work, you pivot to the next project until something breaks through.
Yeah. I guess that project's exciting because I guess the overarching theme or the goal is to have a plug-and-play diagnostic. So we use this principle called binding-induced DNA assembly. It's kind of like this if-then statement. You give two proteins, two DARPins that can bind to a target independently, and then we have DNA molecules that are attached. So if the binders or the DARPins are binding to the target, then the DNA molecules can come together. Then we can amplify it and detect it. So in theory, once you can figure out the DNA part, you don't have to change anything else. You just switch the binders, switch your DARPins. And so you can detect Ebola or SARS-CoV-2, the C. diff toxins or the Shiga toxins. And so that's like a universal application. But the part that we're really working on right now is to I guess, make it more point of care. So just remove the PCR block entirely and just start amplifying it at just one temperature, so like 65 Celsius. Just have like the patient or whatever, whoever, wherever they're at, just put it on a heat block and then.
So this could be like maybe like an urgent care application, or maybe not even in urgent care, maybe in like contexts where there's not even like maybe an office or something like that.
So like maybe even the goal, you had said stuff about how the people that typically get Shiga toxin are in low resource areas. So would it be something that you mainly want to implement in those types of areas, or is it useful in like a wider clinical setting in areas with more resources? Like what's the main push for the?
I think for, I guess, places that are not resource intensive, I think 2 million, around 2 million to 3 million people in the U.S. every year get infected with Shiga toxin producing E. coli, and then even more globally. So it's, I guess, it's applicable worldwide, but I guess in the U.S. we have more opportunities to go to some kind of clinic. But I think worldwide will be nice. Because I guess one of the goals for this would be to make a kit that can be multiplexed, meaning that we have multiple different probes. Or I guess we're maybe probing to detect SARS-CoV-2, or maybe we're probing to detect maybe a flu virus that's not SARS-CoV-2, or maybe we're trying to detect some kind of other respiratory virus and then figure out which one. Because sometimes when you sneeze, you're like, or you're coughing, you're like, do I have COVID? Is it allergies? Like, what do I have? So just to be able to discern that would be pretty nice.
Do you think it'd be possible to have, and maybe it's not based off of the system that you're describing, but is it possible that you could have just like maybe like one saliva sample, like kind of like what we do for like COVID tests, and you could test like a variety of things like Shiga toxin, COVID, like a number of things that they might commonly want to look for that could create symptoms that kind of overlap?
I guess it would depend on where the target is. Because I think Shiga toxin is not in your saliva, it's in the blood and stool. And then for SARS-CoV, yeah. I guess it would depend on how you would multiplex it.
Right.
So you could get a couple different samples, whether it would be saliva, blood, what have you, and then run a couple tests that test for multiple things, is what you're saying?
Yeah, that'd be cool. That'd be cool. But that's a long way. That's from now.
Many clinical trials later.
Yeah.
Do you hope to pursue that type of work past your PhD in terms of kind of continuing, I know it's a big question, but continuing doing diagnostic type work or pharmaceutical type work?
Yeah, I mean, if we can keep progressing, then I would definitely want to keep pursuing that. But I don't know how all that stuff works right now. Like working with the A&M lawyers and all that, it's a whole tunnel of like of patents and
patent law and all that.
Yeah, we'll see. But it'll be exciting. That'll be cool.
So I guess maybe we'll switch over to something else, but I have one last question about the protein engineering stuff. Of like, you know, obviously we're graduate students, we're reading a lot, we're trying to figure out what's going on in our field and trying to apply that to our research, but what's maybe one of the more interesting things or promising things that you've read recently, like in protein engineering or in diagnostics, like point of care diagnostics, that you think is like really impactful? Maybe like will be like the next big thing. We'll see it in urgent cares everywhere.
Ooh.
Or outside of urgent cares everywhere.
The one that rings a bell immediately, Fanzer proteins.
What's a Fanzer protein?
You've heard of CRISPR-Cas9 gene editing, CRISPR-Cas9 based gene editing.
Okay.
Then you have, I guess recently there was the Omega, I guess another RNA guided endonuclease. I guess the Omega systems. They're like, I guess the protein's called TMP. I guess that works in, I think it's not Archaea, because Archaea has like the CRISPR-Cas12, I believe, and then TMPs are in something else. But they're trying to find a new, I guess, RNA-guided endonuclease within eukaryotes rather than prokaryotes and Archaea and all that. And so they started comparing like TMP and CRISPR-Cas12a, comparing the homology of those, and then they did some like, I guess, bioinformatics to see, I guess they found this protein called Fanzer, and it was discovered in 2013, but they didn't really have like a purpose for it. Kind of like how they found CRISPR-Cas9. They didn't really
Yeah.
know what the heck was going on.
Yeah.
And they started like doing like, I guess, some of these studies, I guess, looking at like the homology of that protein. And it was much smaller than CRISPR-Cas12a. That one was about like 1,300 amino acids, where like the fans, there's like maybe 500, 600 amino acids. So like almost like 3 times smaller. And then I guess they used it for gene editing. And it was able to induce indels. And then they looked at the guide RNA too, because you need a guide RNA to help instruct this endonuclease. And then the structure of that RNA looked just like the CRISPR-Cas12a, which is like insane to think about, because I guess we try to optimize CRISPR-Cas9 to put in humans, or these other, I guess, CRISPR-Cas12a to use in humans, or vice versa. And then we already have like this eukaryotic system, I guess not necessarily in humans, but like in mollusks or these other different organisms that are all eukaryotic. It'll be interesting to see what happens in a couple of years. Because this paper was like in July, so it was relatively recent.
Fresh out.
Did they are they looking at this for like therapeutic purposes, like in humans? Why are they looking specifically in eukaryotes? I'm assuming it's probably something like that, right?
I don't know. I think they found the protein and then they started digging into it, and like they definitely were implying that they can use it for gene editing in humans. I'm not a gene editor myself. I know one of the panel judges here, or the panelists.
It wouldn't be me. I'm a mouser.
I've never heard of Pfanzer, so I don't know. I'm not familiar with it, so I can't say anything about it. I work a lot with CRISPR, but
That could be our next big
Cas9.
thing.
It could be, yeah. It'd be really cool. That's cool. I haven't heard of that. So I have a question. So in an interdisciplinary program, so you're in genetics, but your department that your lab is in is microbial pathogenesis and immunology. How important has collaboration been for your project? You're coming from a totally different background from a lot of people in your department where you're working. I'm imagining that like you're working with maybe some other people in your department or other people from genetics.
I don't think so. For me right now, I guess my lab members do. For my stuff specifically for diagnostics, I don't really know too many. I know for I guess that's not true. For one collaboration with the Shiga toxin stuff, there's a guy in the engineering department who works with I'm not even sure what the technology called. It's like Ramachan spectroscopy.
A spectroscopy?
Something like that with Ramachan in the front, and I was like,
What is a Ramatron?
Some kind of like complicated physics. I'm not sure. I probably butchered the name, but he's using that he wants to use that on the final byproduct of the DNA structure that's formed. But we haven't been in contact with him in a minute. And then there's another group for the AI project. They took our evolution data, right? So I guess to evolve a protein, you take several different rounds of evolution. You challenge it with different buffers to see if it can bind the target and stay on. Is it a strong binder? Is it a non-specific binder? You just throw all this washes, and that's one round, and you do it again, second round. We have all that evolution data and all the mutations that may have survived in every round, and then they put it all on this map. So they did the coding for that. So that's one collaborator. So they asked, this is a website, and they have all the evolution dots. And it kind of just shows you after each round what mutations are favored. That's not too exciting, but it feeds into that AI algorithm for their mutations that they recommend.
I see.
So then we can connect the two dots. And I guess the people who made that algorithm, that's another collaborator technically. So then that's like 2 collaborators, and then we purify it and validate. And so it's a pretty...
So that collaborator that you mentioned with the very fancy spectroscopy, that's for later down the line, right? So is that more for the stage of purification or what, like, because when I think of spectroscopy, I think of characterizing a compound. So is it like, what's their, I guess, bigger goal in helping y'all? Like what role do they play in terms of getting these things eventually to that point of care, that urgent care? Or do you know?
Yeah, that's with the heat block that I guess I mentioned. So you do your structure, whatever, the structure forms for the DNA. The binders bind, the DNA comes together, it forms a structure. You put it on the heat block. It makes a lot of DNA because we put this polymerase in there that helps the amplification of a lot of DNA. But then the question is, how do you visualize it? Are you going to put a dye? And then that's where he will come in with the Yeah, the Ramachan. He'll come in with that.
Sounds like a transformer.
Oh, okay,
The
I
science
see.
transformer.
Okay.
So you'll put some like probes in there to help things light up once a lot of DNA forms.
You could probably do that with just a normal spectrophotometer, right? Because if it's going to be like creating, if the polymerase is going to be
I think he has some
like
binding
synthesizing
probes,
more.
but
Oh, okay.
we'll see.
Well, outside the lab, Ben, you're pretty involved. You do a lot. I know for the Genetics Grad Student Association, Ben, you're the social chair. Could you talk a little bit about what that's like?
Oh, being the
Setting up
social
socials
Yeah.
and hosting events for everybody to have a good time, to get out of the lab and have some fun.
I think I took that position back in, I think it was like June or May, maybe June.
Elected.
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 I think it's been pretty chill so far. You know,
It's a democratic process, Ben.
I'm doing my best to.
You gotta throw that in there that you were chosen
I was chosen.
Not a dictator.
for your social skills.
I took it. We took the throne. Yes. I don't know. It's pretty good so far. I think it's a nice way to get people out of the lab. I'm trying to do things that are like different from the past years, but keep some things from the past years that people are comfortable with, and because College Station can get a little boring, so try to take advantage of what the genetics and genomics program gives us, and then try to take the full opportunity to take advantage. So far, we've been fishing, which is really, really fun, crazy. We got Dr. Threadgill out there. That was amazing. Oh, man. And then the next one was like ice skating, which was really fun. Some of the summer interns fell. That was not so fun.
Laughing at their pain.
Oh, no. And then what was the next one after that? There was axe throwing, maybe a brewery. Maybe the next one will be rock climbing.
So what do you do outside of, scheduling social events for all of us? What are the things that you like to do most when you're not in lab or reading? What are some of your hobbies that help you balance life?
Ooh, there's a lot. I like to watch movies. I go to the theater quite frequently. I like to dance. I'm like officially a member of the Salsa Fusion Club. We do bachata, cumbia, salsa, merengue, a little bit of everything, some sauce.
He's a man of culture.
Oh,
I like to fish a lot, as you know. I like to fish. I used to like lake fishing, but I think sea fishing is really, really get them big monsters out the water. I like to bake, lift, basketball, lots of basketball.
So basically, Ben's a Renaissance man. Does a little bit of everything.
yeah, you didn't put that music career in there.
Yeah, where's the music?
Yeah.
Be shrimp.
Be shrimp on YouTube.
You got science-themed music on there.
Yeah, I got some songs shouting out GGSA. I got a mitochondria song now that recently dropped.
Shameless plug.
Shameless
for the powerhouse
plug.
of the cell.
Facts.
So I got a question for you that might be able to help some of the grad students out there, or aspiring grad students. You're involved in GGSA and our Grad Student Association. Obviously, you have the research going on. A lot of projects, the research going on. It sounds like you got a lot of hobbies outside of the lab. How do you balance all of those? How do you find the time to do everything and do everything well?
I honestly don't know.
You don't have any strategies or pieces of advice you can impart on a hopeful graduate student?
What do you have for the youngins, Ben?
I think for this week specifically, what I found to be really nice was just to wake up at like 5:30, come into lab. I blast my music. That's like an incentive to be there. I have a big boom box, and then I type, read, get the protocols ready, start the experiments. My mentees come in. I have them set up on their stuff. My mentees are basically my second hands. So I'm just like, I can just sit back and then write the stuff I need to write up. And then I guess if you clock out around 3 or 4 because you came in so early, and then it takes a little bit of caffeine sometimes if you're really tired. But I don't know. I think you just gotta make it work. We may get a little power nap, maybe 20 minutes.
So you're also mentoring undergrads. So what's that like? How do you think that's helped you, like in your graduate school endeavors?
I think I started getting undergrads this January. Yeah, I started out with one. She was very helpful, just getting because protein purification, it can be challenging, but if it's like simple, like DARPins to make, I guess they're optimized to be easy to purify, and we have a lot of them to purify. So if you can get some undergrads to do that, that's really great. And then I was like, okay, well, I got one undergrad that's good at purification, then I brought in another undergrad, have that undergrad train them at purification, and then take that original undergrad to have them work on something else, which is like click chemistry, which is some fancy form of chemistry. Have her work on that, and then she gets good at that, and then the cycle kind of continues when I bring in another undergrad, and then have them work on I don't know, some kind of detection system. And that's how I've been doing that so far, but took a lot of time to start it up, but yeah.
So I'm really interested in like everyone's kind of approach to teaching undergrads, because it can be like, as a graduate student with your own project and like everything that you're doing, it could be like really draining. I love my undergrads. They're a really fun time. I love teaching, but an 8-hour day feels like a 16-hour day at the end of it, because you're just doing so much and explaining so much. What's like your philosophy for how you try to like teach them? Have you found the most efficient way to teach your undergrads that you could maybe share some of your teaching hacks with everyone?
It depends on who they are. So when I bring them, I guess I interviewed, for my first one, I interviewed like a panel of 40 people. I guess I interviewed 10 specifically, looked through 40. And then however they are in the first day, the first couple of days, I'm like, okay. And then maybe if I'm explaining something, how much do they pick up that next time I like kind of ask them about it? And then I'm like, if I see that they like puzzles and they like the challenge, I give them a protocol to fill out. And then I'm like all right, figure it out. And then I leave. But if they need a little bit more like guidance, then I'm like, those are the ones I'll stick around with more. Yeah, I think the ones that are like really detail oriented, they catch me on my own details. Those are the ones that I'm gonna have like maybe taking more of the challenge of making their own protocol.
Okay.
It's very tactical. It's like chess. I'm doing chess.
They're all pawns in Ben's
Oh my
plot.
gosh.
All his undergrads. I know.
How do you get the ones that might need more help? How do you get them to that level of independence that some of your students come in just kind of naturally having? What types of like motivational things or strategies do you use to kind of get them to push themselves?
That's a good question.
I like to give small projects that have not been done before in the lab. And if they can get it done, it's like a little sense of pride. And then repeat it, and then step it up a little bit higher. And then I also give props, and then we have a snack box in the lab. I think snacks are a good motivator just to come in and...
Yeah, I'm food motivated, I understand.
I just think we create a work environment that's pretty like friendly and jokey. I'm a pretty jokey person. So then it's just a fun place to be at.
If it's not clear.
Yeah. I don't have any secret sauce. No secret sauce.
No secret sauce.
Secret sauce.
I don't have any secret sauce either, but...
Well, Ben, I have a you got any questions?
No, I was just saying I really thought that was cool. I might implement that with some of my undergrads. That's the hardest thing for me is when they have trouble being independent after a while.
Yeah.
But yeah, definitely. I mean, we had a snack box too, so.
I guess some of the projects I give them, I don't care if they fail on it. And if they mess up completely, then I'm like, this wasn't even that expensive. Then I slide something a little bit harder and then build up the price of the experiment. But I guess the lab's experiments are very high throughput. So it's not like a mouse work or anything like.
I mean, my students do mouse work, but I mean, it's, you build trust and then they kind of get to that point. But yeah, I struggle with that the most, is just like making sure they feel like they're doing a good job. Because I'm always worried. I'm like, that was me. I was the one that messed up, and I still ended up going to grad school. But like, I don't ever want the undergrads to think they can't do something just because it's hard, But
Absolutely, yeah. I'm gonna I've noticed that, like maybe subliminally, I was thinking a little bit like you get a few students and you notice that there's like a spectrum. It's like sometimes people are really independent, they like pick up really fast, especially working with their hands. And other students, it's like maybe they need to build a little bit more confidence and you have to kind of mentor them more one-on-one and work with them in the lab. So it's interesting that that's like one of the first things you try to do is like feel that out. Because I was, I just got the students and I'm like, okay, well, like we're all going to work on this project. And yeah, so I more learned just that by experience, but haven't consciously thought about that. So that's definitely very good advice for anybody that's mentoring undergraduate students or is thinking about it. That's great advice from Ben. All right, so we're going to try to squeeze some more advice out of you. Oh. One last piece of advice for a student that wants to get into protein engineering. What would you impart to them?
I don't know. In terms of preparation, I don't think you need any. I think you just need to be excited. Just need to have the enthusiasm, because I came in from entomology and then very minimal biochemical techniques. So just to come into this lab, it was a learning curve, but like if you have, because I don't even think the mentoring style, people were not mentoring me like great in the lab, right? Because it was during the COVID years. And then I guess the postdocs were just doing their own thing, trying to get data out and leave. So then I was kind of just like fumbling around. So I think anyone can get into it and try to do it. You just gotta be patient and have the passion. Yeah.
The 3 Ps: patience, problem solving, which is 2 words, and passion. Today's episode was hosted by myself, Joe Romanowski,
and Brittni,
and produced by Anthony Matarazzo and Aaron DeSalvio. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, Twitter, and TikTok at Genetics Podcast. Thanks a lot, Ben. We appreciate your time.